Hello, everybody. Welcome to Global Response to COVID-19. This is the webinar number 20. This meeting is supported by the Panamerican Trauma Society, the American Association for Surgical Trauma, International Relations Committee, the World Coalition for Trauma Care, University of Miami, and Virginia Commonwealth University VCU. Everybody remember this meeting has been recorded and will be posted on the Panamerican Trauma Society website for future people that want to watch it. I'd like to invite Dr. Michael Butanos to introduce the panelists from today's webinar.
Yeah, if you could pull up the list. Great. So hello, it is really my pleasure to introduce our panelists for our session number 20. Starting with our dear friend, Dr. Sandro Rizzoli, Medical Director of Trauma at Hamad General Hospital. He's Editor-in-Chief of the Pan-American Journal of Trauma and co-editor of GEMTAC, and he's hailing us from Qatar. And we have Dr. Camillo Ricordi, Professor and Center Director of the Diabetes Research Institute at the University of Miami Miller School of Medicine from the US. Dr. Lilian Abo, and Professor of Infectious Disease at the University of Miami Miller School of Medicine, and the Chief of Infection Control and Antimicrobial Stewardship at Jackson Health System, also from Miami, Florida, in the US. We have Dr. Ricardo Lima, Professor of Surgery at Federal University of State of Rio de Janeiro, and Head of ICU at the Samaritano Hospital, Rio, Brazil. And we have Dr. Esteban Fuenini, our PTS Treasurer, Director of ATLS in Bolivia, Surgeon and General Director of Fuenini Hospital in Santa Cruz de la Sierra from Bolivia. So welcome everyone for our 20th session. Antonio.
Thank you. Today's speaker is going to be Dr. Carl Schulman, a great friend, Professor of Surgery at the Department of Surgery at the University of Miami, also Executive Dean for Research at Miller School of Medicine, here at University of Miami, Florida. One of my partners at Trauma and Critical Care Division. Dr. Susanne Doblecki-Lewis, Associate Professor of Clinical Medicine, Clinical Director of Infectious Diseases at Miller School of Medicine, here at University of Miami. Dr. Rekha, Rapaka, Ph.D., Assistant Professor of Medicine and Infectious Disease Specialist at the Center of Vaccine Development and Global Health at University of Maryland, Baltimore, USA. Dr. Michael Butanos is from Virginia Commonwealth University, past president of the Pan-American Trauma Society also. So Dr. Esteban, could you please introduce all our partner societies and university institutions that have been with us for 20 weeks discussing COVID-19?
Yes, it's my distinct pleasure. Good afternoon, and thank you for the invitation, Antonio. So this presentation is done through the University of Miami, Ryder's Trauma Center, the Pan-American Trauma Society, World Coalition for Trauma Care, and the American Association for the Surgery of Trauma. And we're joined by all these other partner societies, which represent most of the trauma societies within the Americas and also some around the world.
Thank you, Esteban. During the last 20 weeks, we discussed treatments, new guidelines, protocols, first wave, second wave, the pandemic in Europe, then in Latin America, then worldwide. And now we are going to discuss today a good topic that's going to be the vaccine. So first, research, what you learned, what's working, what did not work, and then what's the next step with the immunization vaccines. So let's start. I'd like to ask my friend, Dr. Carl Schulman, to please start the presentation. Carl.
All right, thank you, Antonio. I'm going to share my screen, put it into presentation mode, and make you all very small. So today I'm going to, in 15 minutes, try and do the impossible, which is give a sort of a broad overview of what's been happening in COVID-19 research and potentially where we're going. And then it'll be a nice segue into the vaccines, which are the next great big hope for COVID. So basically I have some disclosures that, I'm going to talk about a lot of off-label or investigational use of products, mostly in the vein of research studies, and they are not often labeled for the use under clinical discussion for things that I'm going to talk about are still investigational. So really, if you think about it, SARS-CoV-2 should be simple to contain, right? It uses a strategy of moving from host to host before an adaptive immune response emerges. It chooses transmission efficiency over resistance to our immune systems. It exposes rather than hides its key antibody recognition sites. It's very vulnerable to conventional vaccines and passive immunizations. And due to its large genome, it evolves slowly, which means a vaccine that works now or in 2021 will hopefully work in the following years. The current key classes of therapeutics that are under investigation kind of fall into two big categories: antivirals and immunomodulators. I'm sure you all are familiar with the remdesivir success and the dexamethasone success, which I'm going to talk about, but there's so many other inhibitors and immune modulators. But this quote is very timely, where it says, appropriate management strategies for patients with COVID-19 are a rapidly evolving therapeutic challenge, and the optimal agents to treat infection or prevent progression to critical illness remain ill-defined. So I think we've learned a lot, we're doing a lot better, but there's still so much more to learn. If you ever wondered, how these drugs all of a sudden popped up for COVID-19, just a little background is, it's really been through large-scale drug repurposing and high-throughput analysis. Here's an example where they looked initially at 5,600 compounds for activity against COVID-19, and they found 64 compounds would have reasonable activity and 19 compounds with good activity, 90% of which hadn't been previously reported as active against COVID. 37 of them are already active launch drugs, 19 of which are in now phase 1 through 3 trials, and 10 in preclinical trials. So this is really where this information comes from. You can see here in the top left, I just wanted to highlight here, remdesivir showed very good dose-dependent inhibition, whereas lopinavir didn't. Again, we know the story, remdesivir seems to be doing well, lopinavir not so well. And then you can see there was initial excitement, as you can see, for azithromycin and hydroxychloroquine on the left-hand side, also good drug-dependent inhibition of COVID-19, but as you can see, or as I'm going to mention, they didn't really pan out as well. So the first big one I'm going to talk about, since it's currently in use clinically and it's still being studied in clinical trials, is remdesivir. So remdesivir was originally given FDA emergency use authorization for severe COVID-19. And just to highlight the definition of severe COVID-19, it's defined as patients with an oxygen saturation less than 94% on room air or requiring supplemental oxygen or mechanical ventilation or ECMO. So that comes up in a lot of the subsequent trials. One of the first big studies was this adaptive study looking at the NIAID study. It's a multi-center adaptive randomized double-blind placebo-controlled phase 3 trial. Basically randomized patients to 10 days of remdesivir versus placebo. And the primary endpoints were time to recovery by day 29 and some other secondary endpoints. They use this clinical status ordinal scale as their endpoint. And you can see number 1 is basically you're not hospitalized. Number 8 is you're dead. And the in-betweens, the 3s, the 4s, the 5s, this is hospitalized patients requiring increasing respiratory care, including ventilation and ECMO. If you look at the baseline characteristics in this trial, the mean age was about in the late 50s here. Most of them were males. It was a fairly long time from symptom onset to randomization, 9 days here. They all, not all, many of them had comorbidities, with over half of them, 50%, having greater than 2 comorbidities, with the most common ones being hypertension, obesity, and type 2 diabetes. So this is very reflective, I think, of the clinical population we're seeing. And this was how they stood out on the baseline ordinal scale. You can see here, this is in the 4 to 7 range. So this is not requiring oxygen all the way up to ECMO. And the majority of them were hospitalized, requiring oxygen or ventilation or ECMO. So the preliminary results after about 1,000 patients after randomization, the thing that's really led this into clinical use is that median recovery time in days went from 15 days to 11 days, highly significant. So it was shortened the time to recovery. You can see here in the subgroup analysis, and I apologize, the slides are busy, but we only have 15 minutes. There were some subgroup differences related to geography in North America, related to race, with white race doing better, related to ethnic group, with not Hispanic or Latino doing better, related to age, with younger groups doing better, and those receiving oxygen doing better. So there were some baseline differences. Sorry, baseline analysis difference. So this was based on their initial characteristics. The second big study that was done with remdesivir was the simple moderate study. So this was patients who weren't quite as sick, and they were randomized to either 5 or 10 days of remdesivir versus standard of care. This was a multi-center randomized open label phase 3 trial. Again, they had to have a moderate COVID-19. The take-home points from this study is that patients who received a shorter 5-day course of remdesivir experienced a similar clinical outcome. More than half of the patients in both treatments groups were discharged by day 14, actually a little bit better in the 5-day treatment group at 64.5% versus 53.8% in the 10-day treatment group. And patients who received remdesivir earlier after symptom onset, certainly within 10 days, had a trend towards improved clinical outcomes. And I think all of the clinicians on the call and the webinar now know that we are working towards getting this started earlier, and it doesn't seem to be as effective later. And there were no new safety signals. So summarizing this, just recently in an article published, an interview article in JAMA, Dr. Anthony Fauci said on remdesivir, it's really a solid single, but not a home run. You know, he's a big baseball fan. He threw out the first pitch at the first Nationals game. So it's very good. It's a piece of armamentarium, but not really a home run. We haven't really seen mortality benefit yet. The other thing that's being used widely now, as you all know, is dexamethasone. You know, dexamethasone is a classic corticosteroid with its classic anti-inflammatory effects that's been used to treat a whole host of diseases. It comes IV and orally. It has all the warnings and side effects that come along with a corticosteroid. The big trial that sort of led to its widespread adoption, I think, is the recovery trial. This was a trial in hospitalized patients with clinical, suspected, or laboratory confirmed COVID. They were randomized to a usual care plus no additional treatment or several other treatment groups, including dexamethasone. So it was a factorial design with simultaneous allocation to one of those treatments, the common things you've heard about, versus convalescent plasma. And if they progressed, they were able to get additional treatment with tocilizumab. They enrolled over 11,000 patients at 175 hospitals in the UK, but in June 8th, the recruitment to the dexamethasone arm was halted because they had sufficient patient numbers to establish a potential benefit. Again, looking at the patient population, mean age was again in the mid to late 60s. Most of these were males. The median time from symptom onset to randomization, again, was about 8 or 9 days. And many of them had the usual comorbidities here listed as diabetes, heart disease, and chronic lung disease distributed equally across the various treatment groups and enrollment groups. What was interesting from this trial and what has been taken in the now standard of care, I think, for COVID is across all comers, dexamethasone had a statistically significant mortality benefit compared to usual care. On the right-hand side here, you see in those patients that were receiving no oxygen, not very sick, that difference was not statistically significant. However, in the patients receiving oxygen only, it was statistically significant. On the left here you see, and in patients receiving invasive mechanical ventilation, it was perhaps even more pronounced, the effect of the mortality benefit. So it suggests this is better in the later stages when you've got that severe inflammatory response already going from the COVID-19, that the steroids are helpful at that phase of the trial. And I think most of the clinical protocols nowadays are mimicking this with remdesivir early and dexamethasone later. The big bulleted point on this slide is that this data suggests that 1 death is prevented for treatment of every 8 ventilated patients or every 25 patients requiring oxygenation alone. That is a pretty good bang for your buck, considering that dexamethasone is fairly well tolerated, and although it has the side effects of a steroid, they are not widespread. Interestingly, the World Health Organization and their guidelines for dexamethasone, I checked this just yesterday, even though this slide says June 25th, says the same thing on their website today, that the WHO is still in the process of updating treatment guidelines to include dexamethasone or other steroids. Whereas I think around the country and certainly around the world, and certainly here in Miami, we've already initiated this. So now I'm going to finish up with a few minutes on the newer investigational treatments you probably have heard some about, maybe not about others. But before that, if you look at this slide, this is very interesting. So this was the geographic distribution of the clinical trials for COVID-19 in the very early phases. You can see from January to February and all the way up into the beginning of March, it was almost all in China. Naturally, that's where the cases were. As it started to spread across the rest of the world, you can see the USA and the Europe in the very last column here, March 16th, started to come on strong in terms of where the clinical trials were happening. You have to have the patients. And then this was downloaded just today. just recently came out a couple of weeks ago, and I highlighted some of the things because they're hard to read, but this is the total amount of interventional studies currently occurring across the world. And you can see now the United States has the most number of trials, followed by China, France, Iran, Spain, and Italy. So obviously the clinical trials follow where the disease is. We wish it weren't the case in the United States, but we're doing our best to understand this disease as it continues to show itself. A couple of other things that may have not gotten such worldwide news are stem cells, and we have Dr. Rekha on the panel today and can talk about his work. But basically, mesenchymal stem cells, there have been several trials, particularly here at the University of Miami, but across the US, because we all know mesenchymal stem cells are anti-inflammatory, they're immunomodulatory, and they're actually a good match, I think, for the hyperinflammatory state of COVID-19. We have done 3 trials already here at the University of Miami, one with moderate COVID-19 disease, one with more severe disease with the patients on the ventilator. That's Dr. Rekha's study from the Diabetes Research Institute. And then also looking at those on ECMO. And we've done this with our Interdisciplinary Stem Cell Institute and with our Diabetes Research Institute. And 2 of these trials are now passing from their phase 1 trial, which was very successful, tremendous safety profile, and now working into phase 2 and phase 3 studies to try and show efficacy. I can tell you preliminarily that there was good trends towards efficacy, but none of the early trials, of course, were powered to show that. Another trial we have going here is Viptidil. This is a vasoactive intestinal polypeptide. It's a short half-life neuropeptide. It was originally given an IND back in 1996 for ARDS, so you can imagine that it's widely popular thinking we could use it for COVID-19. And this is currently undergoing study here for patients on the ventilator with COVID-19. And one of the last things will be convalescent plasma. This is probably the biggest thing in the news lately. Everybody's being encouraged to donate. So as you all know, when you recover from COVID-19, most patients will have antibodies. They can then donate blood, and those neutralizing antibodies in their plasma can be given to patients either for prophylaxis or for therapy after they've got the disease. And there are studies currently underway in both of these realms. This initial study looked at 19 patients with severe COVID-19 that were treated with convalescent plasma. Mean age was 55 years old. And I just want to go to the outcomes. Basically, the preliminary results showed improvements in oxygenation, improvement in other symptoms, radiology, viral load, and patients requiring mechanical ventilation or ECMO seemed to be more successful at being weaned off. But none of these early studies were powered to really tell us whether this works or not, which is why now there's a national effort sponsored by the National Center for Advancing Translational Sciences from the NIH called NCATS, part of their Operation Warp Speed portfolio, where they're looking at convalescent plasma in patients with moderate disease, hospitalized patients, non-ventilated. This is a phase 2 blinded study. Here at UM, we're going to be actively recruiting for this study. And this is for patients with respiratory symptoms within 3 to 7 days from the onset or within 3 days of hospitalization, not on mechanical ventilation. And this study started in NYU in New York. As you can imagine, their numbers have gone down. And now we're trying to get this up and running in Miami and Texas and some of the other hotspots across the country where we can rapidly enroll enough to see if this is really an efficacious treatment. And then this last slide is sort of a summary of the investigational treatments. If you look on the left-hand side, basically remdesivir is currently recommended for hospitalized patients with severe COVID. Again, severe here is requiring oxygen or greater. On the right-hand side, you can see dexamethasone is currently recommended for that same group of patients, and now we know to use one early and one a little later. And for lots of the other studies and things you've heard about, the hydroxychloroquine, the lopinavir, convalescent plasma, immune globulins, the inhibitors of cytokines, they're basically all recommended in the context of clinical trial and should not be given outside of a clinical trial because we just don't know, and some of them have significant side effects. And with that, I'd like to thank you. I have to thank my infectious disease colleague, Dr. Jay, who helped me with a lot of these slides, and actually Dr. Abo as well. So I thank you all, and I'd answer questions at the end with the rest of the panelists.
Thank you, Carl. Great presentation. The next speakers are going to be, they were very difficult to get. So you know they're very busy. That's vaccine time trials. And Dr. Susanne Doblecki-Lewis, I have been trying to have her here for a couple of weeks already. I finally got her. And thank you, Susanne, for canceling some meetings or working around. I know that Your time has been very valuable, and I'm looking forward to listen to your presentation. Thank you.
Thank you, Dr. Martos. I'm going to share my screen, and thank you so much for inviting me to present at this 20th anniversary conference. I think this is a great conference and learning a lot. So I am going to give a presentation about clinical trials and how we're proceeding with the vaccine trials. I'm not going to present a lot of data. Oops, let me move forward here. I'm just going to give a little bit of an overview of how we're thinking about vaccine trials in this very accelerated time frame that we've been given during COVID-19. And as many of you know, this is part of Operation Warp Speed that Dr. Schulman mentioned, has many aspects, and vaccine is certainly one of the major aspects of that portfolio. So we'll talk a little bit about a framework for how we think about vaccine clinical trials and what are some of the important considerations. I think, most of us are seeing every day in the news, in the New York Times, on CNN, more and more data coming out about all of the vaccine candidates. So I thought it would be useful to go through what are some of our considerations and how we evaluate these, and considerations also in designing trials and executing them. So you may have seen this. This was in the New York Times, and I think it's really useful. We all know that vaccine development right now is very accelerated, and I think it's useful to go back and see how non-accelerated it typically is. So the typical vaccine timeline takes, from 12 to 16 years, many years of academic research, and then once something looks promising, it goes into preclinical or animal trials, followed by small numbers of candidates looking at safety primarily, and then a larger number of folks usually doing dose ranging and additional safety assessments. And only then after, typically almost, 8-10 years, will the vaccine move into large phase 3 trials. And then after that, it may be licensed, and then they get around to building factories, manufacturing, and distributing a vaccine. And all of that takes a lot of time too. And what's happening now is really accelerated, obviously. And I think it's important to recognize that the way that it's accelerated is really just overlapping these trials. So, I think here in the US, at least, it's important to recognize that we're not actually skipping phases. We can talk about Russia and their Sputnik 5 vaccine, which they've released without any phase 3 data. But our typical vaccine development would involve all of these phases, and after phase 3, which are the many thousand person trials, safety and efficacy, if established, would lead to licensing of the vaccine. What's really accelerated here is the building of the factories, manufacturing of the vaccines before they get to phase 3. So we've seen that happen with multiple companies in the United States, with the government buying hundreds of millions of doses of vaccine without yet knowing if it's effective, and really, effectively hedging their bets. This is the way that we're harmonizing the protocols across multiple platforms. The plan is for 5 platforms right now, and using the clinical trials networks that are already existing for HIV vaccine, HIV prevention, in some cases HIV treatment, and other infectious diseases, primarily in the United States, but not entirely. So there are also research units in South America and Africa, and also in Asia that are collaborating on these in some cases, and not all of the trials, but in many of them. Also harmonized data and safety monitoring boards, and looking at similar endpoints and similarly comparable statistical analyses. And as I mentioned, there are 5 phase 3 trials that are planned so far in collaboration with industry. I've listed them there. And I realize I didn't finish filling out all of their mechanisms. So several of them are adenovirus based. The Moderna trial, which is the one that has, the first one that has started of these 5, is an mRNA based vaccine. This would be the first mRNA vaccine that would be licensed for humans if it's successful. And Novavax and Sanofi's Novavax is a protein-based vaccine, all of them focusing really on the spike protein as their primary antigen. Each trial will have 30,000 participants. So you can see that even just within the co-VPN planned studies. This is a huge number of participants across all of the trials. And like I said, the goal is to have a harmonized protocol. So what do we look for vaccine-induced protection? I think it's important to distinguish between correlates of immunity, in other words, signals that someone may be protected from SARS-CoV-2 or COVID-19 disease. So this could be, and we often talk about levels of neutralizing antibodies, but it's quite unclear what exact levels would be useful, if this is the best endpoint at all. And as we all know, there's also many questions about how these neutralizing antibodies, if they're present, decline over time. And we're seeing that in some cases they can decline rather quickly. And some people, even those who have proven COVID disease, may have relatively low levels of neutralizing antibodies. There are also binding antibodies which bind and do not neutralize. You know, there's some concern about these, and that they may, in some cases, cause exacerbation of inflammation, theoretically at least. It has not been seen in in the animal models and the studies to date. In the presence of immune cells, so there's getting a lot more attention now, T-cell immunity, and the importance of T-cell signatures, especially Th1 biased signatures, and we often detect that by cytokines. And then, of course, there's many questions about if there is immunity, if there is protection from disease, how long does it last, and from what disease? And I mean by that, especially with respiratory viruses, it's frequently very difficult to get sterilizing immunity, or especially immunity in the upper respiratory tract. So one possible outcome from our vaccine trials is that we may have a vaccine that is relatively protective from lower respiratory tract disease and severe disease and pneumonia, but still allows viral replication in the upper respiratory tract, which may lead to ongoing transmission, even though it's protective to more severe disease. And again, when we're talking about mechanisms of immunity, it's usually complex. So, and we can't measure all of them. I just mentioned sterilizing immunity and how it can be difficult, and how mucosal surfaces can have very different immunological milieu from blood, where we're often sampling for, or from tissue. So it's really difficult to get at these more subtle differences in mechanisms that may profoundly impact how effective a vaccine is, or for which disease state. So when we think about how to do vaccine endpoints, most of the trials that I mentioned, and all of them really, are focusing on clinically symptomatic disease. So what we're looking for, people who have symptoms of COVID, who then go on to have PCR proven disease. So it's not surveillance by labs, it's surveillance for symptoms. This requires a large number of participants, because as we know, many cases are asymptomatic, and so those won't count towards your endpoint. And of course, you can also evaluate severe disease as a subgroup, which is helpful, because a vaccine that could prevent against severe disease, even if it didn't avert all transmissions, could be very useful. And again, you know, there's different endpoints for transmissible disease versus clinically severe disease. And infection or seroconversion is a secondary endpoint for these studies. But you do need to have a really good idea of general incidence in the community, and to standardize assays across laboratories and studies, and those things can be difficult to do. And it's also can be challenging, because many of these vaccines can also cause seroconversion, as can disease, and you have to be careful to have laboratory assays that can distinguish between the two. So safety concerns with vaccines, you know, obviously there can be anaphylaxis or immediate adverse events. So far in the vaccines that have been tested, this has not been a large issue. Autoimmune sequelae are something that people do talk about and worry about. And, you know, I think these are reasons to look over time at vaccines and not have short-term endpoints. There's live attenuated vaccines are not being developed for this virus. But, you know, people do worry when you think about accelerating vaccine development about some of the things that have happened in the past, for instance, with an inactivated disease vaccine that had a failure to inactivate, causing, you know, polio disease in 1955, when there was a rush to develop that vaccine. So I think these are things that people worry about when they hear that we're working very fast to develop a vaccine, because historically, you know, not everything readily apparent during the phase 1 and phase 2 trials, and it is important to look at larger groups over long periods of time. And there's also the concern at this point really is theoretical for SARS-CoV, but that antibodies that bind without neutralizing can cause worse disease with an infection or reinfection event by increasing the formation of immune complexes or biasing towards Th2 responses. So this is something that's being looked at very carefully. In the early vaccine development, there's a lot of concern and attention paid to what kind of T-cell responses have been elicited. And for the vaccines that are going forward, it has been Th1 biased, which is reassuring. And another concern, especially to people who are receiving the vaccine, are reactogenicity. So these are the things that you feel after you get a vaccine. We all, feel pain, redness, swelling frequently with the vaccines that are already commercially available and that we've all received. And these can vary based on many, many different factors, the vaccine, but also intrinsic factors of the people who are receiving the vaccines and their own experience of the day, really. So depending on how people are feeling when they go in to get a vaccine can really impact how they react to it. But some vaccines are more reactogenic than others, and this can really impact how readily the public is willing to accept a vaccine. And sometimes there's a thought that there's a balance between the necessary stimulation to elicit a response, but there's not always a correlation. So it's not always no pain, no gain. Sometimes those things are really not correlated. And another consideration is manufacturing and distribution. So some of these are easier to make than others. And the speed and flexibility of manufacturing, how difficult it is to scale and cost, and how much it costs to manufacture, and the stability and cold chain requirements are all critical decisions about what vaccine candidates to bring forward. And this can vary based on area of the world. It can vary based on how remote folks are from central facilities. And it may be that there are different vaccines that are more appropriate for some settings than for others. And as we're thinking about these trials, I think it's also critically important to think about who will be enrolled in these trials. And it's extremely important that we have broad representation of the diverse communities who are at risk for COVID-19 and at risk for progression to severe COVID-19. So there's a big effort with the CoVPN trials to really try to do a deep community recruitment to ensure that we are getting that representation to ensure generalizability and public health impact of a vaccine once it's eventually approved. So thank you very much. We are participating here in the CoVPN network. There's a national registry, so I know folks are on the video from all over. And if you're in the US, going to the National Participant Registry allows registration for all of the prevention trials, not just the vaccine, also monoclonal antibodies and future vaccine trials. Thank you.
Thank you very much, Dr. Doblecki. Great presentation. You already have some questions, so for sure we're going to have great discussions in some moments. Now let's go for the last presentation, Dr. Rekha Rapaka from University of Maryland. Thank you very much for making the time to present today. We're looking forward for a presentation and we're going to learn a lot, and then we're going to have a lot of discussions after that. Thank you very much.
All right, can you all hear me?
Yes.
Okay, wonderful. Thank you so much for the opportunity to speak today. So I'm presenting from the University of Maryland. I'm an adult infectious disease physician and immunologist, and I too will be discussing vaccines that are where we stand currently. So just some disclosures. I'm a co-investigator on the Pfizer COVID-19 mRNA vaccine trial, and I will be presenting and discussing some investigational products in some detail. So just a brief outline, and some of this overlaps a little bit with Dr. Doblecki's talk, so I'll kind of move over that quickly. But big picture questions on vaccine design during a pandemic. How do we approach? Then getting into a little more of the nitty gritty on SARS-CoV-2, vaccines in particular, questions and aspects that relate to their development, and kind of a snapshot of the current candidates, the main candidates. And then I'll go into a little bit of detail on the COVID-19 vaccine clinical trial findings that are of some of the leading candidates that are in stage 3 development. So as was mentioned very nicely in the last talk, so there's usually a really detailed process for traditional vaccine development. It's a multi-year process, and it's done that way for a reason, but we're in a pandemic, so we've modified our strategies. The phases are now overlapping, and at this point, as of today, we have 165 candidates, 40 in clinical trials currently, and 8 candidates are in phase 3 trials as of August 11th. So there's basically yesterday. So basically we're moving very fast. We've just started the first human trials, in March, and we've now vaccinated hundreds of thousands of people with vaccines, and we are learning about them every day very quickly. So even though we are moving at a rapid pace, we are still using the same principles of vaccine design. First, the critical aspect of identifying the ideal target. And so we've rapidly learned a lot about the viral structure, the composition of SARS-CoV-2, and the process of infection. There's still much to learn, but we have some basics that have informed strategies. We also are using a lot of our inferences on the immunological mechanisms of host defense, first against similar respiratory viruses in the same family, so SARS-CoV-1 and MERS-CoV. The second kind of pillar is how we target how we basically package this target. And so that really involves, questions about what's the optimal delivery platform. Now we're using some really exciting new platforms, which have been alluded to. I'll discuss in a little more detail further. What adjuvants could potentially really lead us to the right immune responses, the ones we want. So as mentioned earlier, we kind of are seeking a Th1-based or a certain T-cell helper response. We want to get lots of antibodies with good isotypes and effector functions. And that can be done with adjuvants. There's variations in dosing regimen that impact practicality, but also impact the quality of the immune response. And then even routes of immunization can impact significantly the quality of the immune response. And then thirdly, as is also just so key, the populations that we're studying and how we deliver vaccine is so important. We're looking at safety, tolerability, the quality of the immune responses that we are develop on in these vaccines, and then the importance truly of understanding the longevity of immune responses. We want to develop vaccines that ultimately provide immunologic memory and are efficacious, of course. And then finally, I think this pandemic has led us to also question, how do we really guard these very unique populations that, for example, are susceptible or at higher risk of severe disease, such as individuals with history of organ transplants, people with HIV infection, cancer patients, older patients who have immunosenescence related to that's age dependent. So these are the same exact principles, it's just we're moving faster. So what do we know about the pathogen and the immune response? So we know that there are 4 major structural proteins. The spike glycoprotein, which contains the receptor binding domain mediating cell entry. That receptor is the ACE2 receptor. It's found in multiple tissues. It's found in the lung, in the mucosa of the nasopharynx. The other proteins include the M protein, envelope protein. These proteins have been in studies with SARS-CoV-1 and MERS-CoV been targets of antibody-based studies. And then the nucleocapsid protein, which is associated with the RNA of the virus. So as I've kind of already alluded to, the spike protein, and has been discussed earlier already, spike protein is really the primary target right now for these early vaccines. And the spike protein, there's been a lot of really elegant studies looking at how this protein, the biophysical properties of this protein as it interacts with cells and how it changes, and all of that science, how that actually would impact the way we design a vaccine. So for example, the mRNA platforms that are being used, the mRNA sequence is being adjusted so that for some of these vaccines, we are looking at the pre-fusion form of the spike protein and designing an antibody response with the vaccine against that. Neutralizing antibodies against spike protein are protective in SARS-CoV-1 and MERS-CoV. And so they've been shown to impair infection of cells in vitro in SARS-CoV-2. So this is why there's been so much emphasis on this particular protein. So this is just a list of the some of the leading candidates. There are 8 vaccines that are currently in phase 3 trials. I'm just mentioning the ones that I'm the most familiar with and that are the most well known. I bolded the 4 that we'll talk about in just a little bit of detail regarding the findings, but I wanted to emphasize, the diversity of platforms. So we really have never, as was mentioned, RNA-based vaccines have really, for the first time, been used in humans in the setting of designing a vaccine for SARS-CoV-2. There are non-replicating viral vectors, and we do have some precedent for use of adenovirus-based vectors in a licensed vaccine with Ebola. We are also looking at the traditional kinds of vaccines. So there are groups that are making protein subunit-based vaccines. There is a group working on inactivated whole virus vaccines with adjuvants. And then there are strategies involving DNA-based vaccines. So, and as I mentioned, these are all essentially targeting the RNA, DNA, protein subunit, and viral vector at this point, or targeting the spike or a component of the spike protein. So just to give a very brief overview of this concept of DNA and RNA-based vaccination, since it's not used clinically and it's really been more experimental for a long time. So with the DNA-based vaccine strategy that's going on currently for coronavirus, essentially the spike gene is put into a plasmid. And then, the way these nucleic acids are presented, they have to actually enter into the cell and go to a place where they can be efficacious. So the DNA has to enter the cell through a process called electroporation, which involves basically an electrical current actually. And then the DNA is uptaken into the cell. Then the DNA is transcribed to RNA, and then the host machinery allows for the protein to basically be made and post-translational modifications to occur. So the antigen looks like what might be seen in infection. With the RNA-based vaccines, the ones that are moving right now involve encasement in a lipid coat. It's called a lipid nanoparticle. And that basically allows for the RNA to kind of safely get to the cell and integrate into the cell. And then that RNA is then translated into protein. And then the proteins are digested. They are taken up by antigen-presenting cells. And then we see immune responses in that context. One of the really exciting aspects of the DNA RNA-based vaccines is that you can make them quickly. So this process is not something that takes very long to scale up. If you found a candidate if you want to manipulate a candidate, you can also do that somewhat easily. So there are really, really great aspects to the DNA RNA-based vaccines. With the viral vector vaccines, there are strategies involving replicated viral vector replicating. So weakened measles virus that contains the coronavirus spike gene, and then that's administered, infects cells, it is replicating, so it's able to continue to make antigen and then provoke an immune response. And then the non-replicating viral vector are, at least in the case of SARS-CoV-2 vaccine design, are adenoviruses containing the current coronavirus spike gene and basically also infecting cells. They cannot replicate, so there's no concern about kind of escape and change. It's basically administered, immune response is generated. And there, the challenges could be, for example, when using a non-replicating adenovirus vector, we have immunity to a lot of different adenoviruses. So you could potentially blunt an immune response with pre-existing antibodies or T cells that exist against adenovirus. Additionally, there's some risk as well considered with using chimp adenoviruses, which is described in some of the trials I'll be describing later. So while the DNA RNA vaccines don't really have a licensed platform, the replicating vectors as well do not, but the adenovirus, yes, in the case of the Ebola vaccine, which was licensed in December of 2019. So now kind of just leaping into the, so the 4 vaccine trials that I thought I would mention. So all of this data has been published in July. One of the papers is still in medRxiv, but basically this just want to give people a flavor of sort of the main vaccines that are moving and ones that you should be looking at to hear more about soon. So the Oxford AstraZeneca COVID-19 vaccine is basically the full-length spike protein expressed on a chimp adenovirus vector. And it was given with a 28-day booster. This was a large trial enrolling over 1,000 adults in the UK. It was multi-center within the UK, and it was single-blinded, so patients aged 18 to 55 were enrolled here. And so for all of these trials, as was alluded to in the last talk, so there's an interest in characterizing the qualities of antibodies to some level. So really, how much antibody is made? Can this antibody that is made against spike or its component actually neutralize virus, or they also use other techniques to basically make virus-like particles that are infectious that can be tested for neutralization. There is also emphasis on looking for T-cell responses. And then of course, characterizing how are people really reacting to this? Is this safe? Is it, you know, what are the side effects people are having? So here, I'm just showing a panel from each of these studies, but this is basically demonstrating a neutralization findings for this group. So basically, you can see that in this panel, individuals are mounting over time by 28 days a pretty significant neutralization titer, but it really enhances with the boost at 28 days. So boosting seems to be important, at least in this vaccine. And they also demonstrate that when they take the peripheral blood and cells from these volunteers and look at the T-cell responses, they see that there are interferon gamma responses that occur. So there are T-cells that are also made that are specific to the spike protein. And they have an interesting angle in their study where they looked at how the kind of adverse events like pain, fever, headache, all of these things that people experience could be limited by acetaminophen, which was started prophylactically. So their trial showed very exciting data, and it's moving from, it's in phase 3 right now, and with plans to enroll 30,000 across Europe. Another trial that's on the move in phase 3, planning to enroll up to 40,000 patients, is based off of the data that was found on this Ad5 vector containing the full-length spike protein. And so this is the CanSino COVID-19 vaccine. And this was published in July in the Lancet. And basically what they showed here is they really just gave one dose and didn't really evaluate with a booster. And they enrolled 500 patients. It was single center, double-blinded, and they enrolled older patients up to age 83. What I'm showing you here, just to give some diversity of the kind of things they were looking at. So they compared, you can see, you know, sort of the baseline T-cell responses on the left, and then with the post-vaccination, you can see a nice induction of T-cell responses that are expressing interferon gamma, and really no difference between the doses, which was nice because actually their higher dose, the 1 × 10 to the 11th viral particles, had significant adverse events. They also evaluated, since this is using Ad5 as the vector, whether baseline antibodies impacted the immune response, and they saw that there really weren't significant changes compared to the other group. So this is also an exciting vaccine on the move. This vaccine is currently in medRxiv, but will be published very soon. And this is one of the mRNA lipid nanoparticles. So this is Pfizer's COVID-19 vaccine, which basically this mRNA is only expressing the receptor binding domain of spike instead of the entire protein. And they compared to normal saline placebo with a 21-day booster. And they also assessed different doses of the mRNA and how that impacted responses. And what I'm showing you here is the serum neutralizing titer on the left, kind of the lowest dose, and then the middle is kind of the medium dose, and then the highest dose was actually very immunogenic and reactive, so that wasn't really studied much for further comparison. But the neutralization response was basically similar to in the middle group, and also the lower dose similar to convalescent serum as far as neutralization. And there's high titers, IgG titers induced against the receptor binding domain. And given this information, the plan is also to enroll, again, up to 30,000 individuals internationally up to age 85. And I saw just the other day that they're also working to incorporate special populations. So they're going to be also offering this vaccine to individuals with HIV infection. The Moderna NIAID COVID-19 vaccine is just the last one I'll focus on. And this is also an mRNA vaccine that the findings were published in New England Journal of Medicine on the 14th of July. And basically this is a modified mRNA lipid nanoparticle containing the spike, but in a particular conformation for which they made some amino acid substitutions to basically stabilize it and have the immune system see it in that conformation. And that was compared against placebo. This was double-blinded. This involved a 28-day booster and was a smaller trial of just 45 adults, but evaluated closely. And again, they characterized many things. I'm showing you the neutralization response at the 25 microgram and 100 microgram dose, and then comparison to convalescent serum. And you can see that there's a response. There are high quantities of these antibodies generated, and the neutralization is comparable or increased compared to convalescent serum. And they too also looked at T helper cell responses. We know these are going to be important for even just generating high affinity antibody responses. And they demonstrated that in the supplemental figures of this paper that T cells are making TNF and interferon gamma, again, kind of moving more with the Th1 helper cell phenotype. And so now we're at the phase where this vaccine is moving to enroll up to 30,000 patients, individuals above age 18, so no upper limit on the age. And as similar to Pfizer's vaccine, also enrolling HIV-infected patients to sort of broaden the scope of potential recipients. So that's kind of my very quick summary, and I just want to say thank you for the opportunity and to speak, and I'm happy to take questions at the end of this session.
Thank you very much. Great presentation. We're going to have a lot to discuss. We're going to go straight to a quick pool of questions. We do this every session to try to understand where the multi-regions in the world are in terms of the epidemic. So Valeria, can you please launch the questions? Please, let's quickly answer that. So number one, does your country area have social distance practice in place right now? You have yes, being respected, yes, less than 50% respected, no. You had, but already in reopening phase, or you don't know. Number two, does a region or area is right now is in lockdown? Okay. Question number three. Question number three. Where does your area? What's happening in your area regarding the curve of positive cases? Still increasing a lot? Slightly increasing? Is plateau? Slightly decreasing? Very decreased and unable to know right now. You know, multiple areas, different areas have different ways right now. Question number four: do you have protocols to treat the COVID and guidelines to treat all the COVID-19 patients, or any physician can do anything you want? Number five, are physicians or healthcare professionals, nurses, everybody at the trauma and ER, wearing N95 masks all the times during patient care? Yes of them, some of them, none of them. Okay. Number six, what's the first line medication that you start early once a patient has diagnosed positive COVID? even he's sent home. So patient has a diagnosis positive, is sent home. What kind of medication treatment do you give to this patient that is positive and you send home? Okay. Number 7. What kind of medication do you give to or treatment do you give to the patient that is sick enough to be admitted to the hospital? Okay, a patient that needs to be admitted to the hospital. What do you give to this patient? Okay, and next question, what kind of treatment do you give to patients that are admitted to intensive care unit? The patients are critical, need to go to intensive care unit. What kind of treatment do you give to these patients? What do you have available? Okay, next question. Pay attention. Let's see. Number 9. What kind of treatment or medication that you use to treat patients do you believe that makes the patient improve? Remember, in the beginning, you give a lot of treatment, a lot of medication. What do you give to the patient that you believe that is helping patients right now? Okay. What kind of last one? What kind of medication are given to your patients, or you gave to your patient at some point that don't believe that is helping the patient, didn't improve the patient situation? Here's the controversy. In the beginning, you use a lot of chloroquine, azithromycin. What do you think that you use and didn't work? Okay. At the end, please click submit, press submit, and then later we're going to discuss the results. Okay. So Dr. Abutanos, please, could you start calling some of our panelists to start discussing, and I have a couple of questions on Q&A. Let's start discussion about these important topics and great presentation that happened today.
So thanks, Antonio. we're gonna look at some of the question and answers that were asked and see if some of our panels can handle some of these questions. One of the questions came in, is the adenoviral vaccine lesser doses than gene therapy trials? So the worry of David Machado is that as adenoviral vector pass through the liver, they can induce failure. Therefore, hence the initial fear of the gene therapy trials. That could be one of the, if any of our speakers or panelists want to handle this question. So we could see if Dr. Schulman's still on and want to see how he could answer that question, or anybody that's Doblecki, anybody else.
Yeah, I would leave that to one of the vaccine experts.
That's what I thought you would.
That's an interesting question. I don't know if he's asking whether the pass through the liver make it inactive or actually induces liver failure. So I'd leave that up to one of the experts.
I think the worry is about the side effect, the liver failure.
I mean, I can make a general comment and then I defer to others. But basically, you know, the gene therapy trials from 15 plus years back, there definitely were problems with dosing of adenovirus and, you know, safety of adenovirus, and the way the trials were specifically run also led often to adverse effects. I think, you know, in the context of replication incompetent virus and its delivery to specific locations, so these are being delivered not really systemically, but really right into the muscle, I think there's less likelihood of really seeing things like that, at least with a replication incompetent virus, where there's evidence as well that after the antigens given, you know, there's not really much, you know, propagation of the virus. It's not possible for it to occur. So I would just say I think that's unlikely from at least the vantage point of the replication and competent viruses. And maybe you could run into issues like that with dosage when you're thinking about the adenovirus or other vectors where you have replication as a possible aspect of its delivery.
Okay.
Yeah, I think I agree. And I think we do have a lot of adenovirus-based vaccines. It's not a new platform. So there is a lot of experience with that with other infectious diseases and prophylactic vaccines. So I think it's less of an issue than in the gene therapy milieu, which is a little bit different.
Okay. I have another question. Actually, one of the presentation, I think Dr. Schulman gave in his presentation mentioned that there is some racial difference when using remdesivir, and that the impact in non-Hispanic was actually lower so actually was better than in Hispanic. I was just wondering if that has any impact on our Latin American colleagues, especially that this is where now we're seeing more of the surge and in the Latin region, and the use of remdesivir. Maybe I just see maybe Esteban, maybe you could address this, or Sandro, or anybody.
But even before that, I just might make a comment that the differences may not be related to the race or the ethnicity, but the social determinants of health, particularly in the area where these trials were carried out, that may be the underlying cause of the differences. So when you go to a population that's completely made up of these type of race and ethnicity, then it may not actually turn out to be an actual difference in how people respond. But just as a note before that goes any further.
Good point.
I do have a comment on that. So we do know that we're seeing an increased rate of severe illness, both in children and adults in Hispanic communities in the United States. Again, and they mix Hispanics on all socioeconomic levels of Hispanics, and the same as in Latin America, in the United States, all Hispanics are not the same. Now, we do know that Hispanics tend to present sicker, and perhaps that also has to do with the response to remdesivir. What we are seeing in clinical practice is that there's a group of patients, even young patients under 50, that are not responding to remdesivir when they're present with cytokine storm and severe illness. We have not completed our analysis. So one thing is the clinical trial, and a different one is your reality and the patient population that you see, especially when people present with advanced illness, not in the early stages when they're minimally symptomatic or very recent infection. So it has to do with the timing of the infection, the viral load of that infection, the environment where that patient lives, their comorbidities, and there might be something, with race that we still don't understand.
Okay.
So, Mike, since you asked me the question, and for the people who don't know me, I'm a surgeon, but I'm in Bolivia. Remdesivir is not available through any clinical trials being held right now in Bolivia. Having said that, people are selling remdesivir, and it is of a questionable authenticity. Since one of the products is coming from Bangladesh, nothing against Bangladesh, but I am not aware, and we investigated that Gilead actually has authorized anybody in Bangladesh to produce it. There has been some other ones that were supposed to go to a Central American country and were somehow diverted to Bolivia and enter through contraband. So those are some of the issues that we're facing, and the question and the concern is, it's a very expensive drug, and we're not sure, first of all, if it's efficacious for the patients, because that's still going through clinical trials, but even worse, people are purchasing an item which may not even be what it claims to be.
Okay. Dr. Lima, is there any difference that you've seen in Brazil with the use of remdesivir?
No. Thanks for the invitation. First, Antonio, thanks a lot. We do not have remdesivir here available in Brazil, and I work in a private hospital. I did not see it with legal or illegal or sometimes like a generic form of that. We do not see that. But we are very worried about, I don't know, you know best than us, but remdesivir was in market since the Ebola, and the impression that sometimes People want to, I used to say there is some drugs that are waiting for a sick or looking for a sick sometime that, and I'm not sure this is the order, the natural order of that. But in Brazil, we do not have experience with remdesivir.
Okay. Before I have Dr. Martos ask a question, but one quick question. So we presented, and that was an excellent presentation on all the vaccine, and very exciting what's to come. I didn't see anything, and this is also one of the questions that my friend Jorge Rabat also was asking. What do we know about the Russian vaccine? What is its impact? Would it have any effect on how the rest of the world will use it? And anybody comment on that, especially from our expert on vaccine?
I don't know if Dr. Rapaka wants to comment. I think we don't know a whole lot about the vaccine. At least I don't, and I'll defer to our colleague on the call. They've said that it's an adenovirus vector-based vaccine, and that's about it. That's about all that I know. And I think it will potentially have an impact. I think there's multiple ways that it could have an impact. They're planning to distribute it to many countries. Apparently, they're producing a large number of doses, and hopefully we don't have a negative impact from a bad outcome that's unexpected based on the lack of large clinical trial data. And I think hopefully it also doesn't cause other countries to start making rash decisions about vaccine. Could be another bad impact. But other than that, and on the scientific level, I'll defer to Dr. Rapaka.
Well, I suppose soon we will have some experience, because today in the news, we have just knew that one of Federation State of Brazil in the south, Paraná, published today in the news that he made a contract with Russia to import vaccine only to that state, to that area of region, Brazil in the south. And they want to begin to use that. I hope it functions, but perhaps soon we'll have some experience or some results about that in practice in the south of Brazil.
I would just add that there's so little known about it. It seems to be a spike protein also targeting, but I don't know exactly if it's a full protein or what, through the delivery mechanism with the adenovirus. I think we're all kind of at a loss because, it's one thing to condense this process. It's another thing to just bypass a step and then, mention that this is efficacious and it's the leading, it's won the race, and here it is. I think that's a challenge that we have, and we may see more of this as we go along. So I think it's our duty to really be on top of the science and really communicating the science as best we can to our communities, our patients, because it's going to get more confusing, I think, as time goes on. And so it's a really great opportunity to do everything we can to just always move with the science, and science always beats gossip, I think that's something I take away from coronavirus times. So.
That depends on the country you're from. So I'm going to refer to Dr. Rizzoli, where there is science and also situational political aspects. So how is the situation in Qatar when it comes to vaccine?
So just a comment on different ethnic patients. We are a very multinational country. In fact, less than 10% of the population are actually from Qatar. 90% come from somewhere else in the world. And another similarity here too was that everybody had virtually the same access to care. And there was no cost limitation. You could give whatever you want, anytime you want, to everybody. And what we saw was not much different. We couldn't pinpoint much of a racial difference between the different groups. So I heard Bangladesh, I heard Pakistani, I heard Indians. We have lots of this. They in fact are most of our population, and they were not different from the people from other parts of the world. So it's very interesting. And sorry, there was another question that I missed. That's it. Thank you very much. The other comment is, I'm not surprised that Brazil is willing to test a Russian vaccine, right? So Brazil is still using hydroxychloroquine when there's clear that most places have stopped using it. So I find it unfortunate. I thought in the 21st century, we wouldn't see any more totalitarian governments. We would see very scientific forward ideas and concepts. And unfortunately, we're still seeing things like this vaccine, for example. It's quite surprising to me.
Great. Antonio?
Yeah, I have a question, a comment and a question based on the question from Dr. David Machado. He's asking here, what would be the target percentage of herd immunity, positive serology to provide community protection? And my comment is the following. During the last sections, you had a lot of some presenters from Latin America present some data. And I have from Ecuador, Quito had a huge spike in the beginning. Now Quito's cases are down, hospitalizations are down, but the other cities are up. No, not Quito, Quito is up, but the other cities are down. In Brazil, Rio, São Paulo had a huge spike, the capital, Manaus, Bahia. And then after the first wave, the numbers went down, and the cities, the states, they're completely controlled, completely down, closed after the reopening, huge spikes out of control. Something very similar to happen in the United States. Like New York had a spike, and like Florida was low. As soon as reopening, Florida exploded, like California exploded, Texas exploded. But New York, after reopening, is still going down. Dr. Abu, for example, there's some data from Brazil, and Dr. Lima can talk about that also. They took some favelas, some communities. There's some favelas that had 300,000 people living in a mountain, you know, like in Rio. And they did serology on this community, and they're finding between 35 and 50%, some cases up to 50% of positive IgG on these people. So do you think that a lot of more people had COVID and they are just asymptomatic, and then right now, even without social distance, that's why these places are not going up again? Like Dr. Lima can say, there's no social distance in a lot of places in Rio, in the favelas, a lot of parties going on. And what's the number? So what the number of for herd immunity using these IgG positive plus the vaccines? How far away some places are from that now they're going to have the vaccines? Because there's no reason why Rio de Janeiro and São Paulo and Manaus are not still peaking. They peak and then are down despite of social distance. I'd like my ID colleagues to make their comments, and then Dr. Lima also, please.
So I'm not as politically correct as my colleagues on the call. I always say what I think. I don't think we're reporting what truly is happening in many parts of the world. And as a Venezuelan, we have big favelas too. We don't have social distancing. We have no testing. And we have the same totalitarian governments that should have been abolished decades ago. So I feel that we don't know the whole truth. I do think that, yes, perhaps that for sure there is asymptomatic transmission, and for sure we're not capturing 100% of their population. So yes, there is a big possibility that in these favelas, even though they have multi-generational families, you know, because the grandmother lives in the same little house, you know, as the grandchildren, you know, and perhaps there's something else that is protecting them. But we also have younger crowds, younger people, you know, many children that perhaps are transmitting but are not getting as ill. And that may happen in many parts of poverty around the world. So that could be one issue. I don't think the story is yet over. Yes, New York has seen a downtrend. We're reopening, schools are reopening, the economy is starting to flourish again. Influenza season is about to start. I feel that it's premature to say that, oh, everything is under control. We already saw the wave. There's going to be no second wave because we're going to have a vaccine. I am cautious, but I am not yet as optimistic. I feel that in Florida, we had a first peak, things came down, people started to party like nonstop, and we saw this rapid transmission with the protest, and now we're just finally seeing the downtrend of this second wave. But this is going to come back up I don't know when, but it will go back up. So I don't think we can keep our guard down. As we've heard, the vaccine trials are going to take still some time. The vaccine is not the elimination or the eradication of this virus. We know it with influenza. We know that with other illnesses. It will help tremendously, and we hope we will find a vaccine that will reduce the mortality, which is what we really need. If the majority of the people get a mild illness, we will overcome this. And also, we haven't discussed in depth the potential mutations and the implications. of the different strains of the virus. So I think we know more than we knew in March, but we don't know the full story yet.
Yeah, Dr. Doblecki and Dr. Rapaka.
Sure. So I guess, I think the concept of herd immunity is number one, a little bit scary, because I don't think any of us really want to get to the end of this through that mechanism. It would involve millions of people dying as a result. But it's also really not that well understood. I mean, we have simple ways to calculate what we would need to get to for herd immunity, and it lands us somewhere 70% protection one way or another, right, through acquired immunity or through a vaccine. But then there's so many different ways to look at heterogeneity and subpopulations, and I think it's really difficult to speculate about how it's intriguing and it's kind of interesting to think about, but I don't think we really understand the dynamics of this virus in populations well at all. And definitely not well enough to make plans on that basis. So, I think it's really just sticking to what we know makes sense and works with policies and distancing at this point. And I'm hoping we don't have to rely on herd immunity unless it's from a vaccine or a mechanism that doesn't result in so much morbidity and mortality as the disease itself.
I agree with that completely. And I would just add that this point you raised about, the virus changing is really fascinating, and it's something that's not talked about very much. This virus, in contrast to a lot of other viruses, this virus does not change quickly. Like, it's not like in the scope at all of like HIV. It's pretty constant. However, there are changes that have been observed that could potentially impact the way we design vaccines. So for example, there's been a mutation that's been identified in the spike protein that kind of forces it into a more rigid conformation. Could that impact the way an mRNA that has been designed to look at another conformation vaccine in a patient that's exposed to that particular virus? Yes, it could. So I think those are the questions that we're going to be moving more towards as we learn more about this virus. And as we see, unfortunately, this infection spread, the virus has potential to change. It has a proofreading enzyme, but it's not completely constant, and there are going to be issues. I know there's even concerns for potential remdesivir resistance that could evolve. So those are things we have to work on and think about, and they have to be part of our plan in trying to fight this infection.
Sure, and Dr. Lima, just adding to your comments. So I saw some report of some case of reinfection in São Paulo, reported by the University of São Paulo. So what's happening in Rio? So what's your vision about what's happening? So a lot of people rely on the vaccines or herd immunity, so should not definitely rely on that, all right?
Yeah, sure. There is some mysteries about this. First, I agree with my colleague that it's not finished yet. We don't know what's going to happen. But we see in Rio different social patterns. I work in a private hospital, was the first to begin to treat these patients. After the middle class comes and the other class. In favela is a mystery. We have some favelas we know there is a parallel state there, another parallel order there, that there is some social distancing and others not. But when I see that, I think about Darwin. I believe that people that's in favela, who are living, who are still alive, they escape from many sickness, something, and then they escape from the health system and some other problems, and perhaps they have a different immunity, something like that. But we don't know. I read some about mathematicians who told us that there is around, I don't know what counts they do, what they think about, but they did that some 30% of people or 70% of people resist to COVID and 30% not. I don't know. I have a microcosmos here in my group of doctors who work in front of COVID. We have 40% of people who get sick, and 6% do not get sick, independent to be working in front of COVID. We don't know what's happened, what's other flus or the vaccines or the, I don't know, the immunity. I can talk about immunity. I have two specialists here, but we don't know what's going to happen. I agree that it's not finished. We are having an endemic picture here. I do not close my unit. I used to have 60 patients here from COVID. Now I have 5. I transfer in a month around 8 or 10 patients. And that was I receive a day. Now I receive a month, but it's endemic in Rio. It's not finished, and we don't know what's going to happen. It's, for God's sake, the virus do not change very fast, but we don't know what's going to happen.
If I could jump on that and address this question, Dr. Ricordi. So do you, so based on what Dr. Lehman just mentioned, do you think we know enough about basically this disease to say that some people have genetic immunity to it and that some don't? And the same thing with regard to the response to the vaccine. Some will respond better, some don't. Just in,
well, yes, I think that there may be genetic difference or blood group, as has been postulated, but also epigenetic differences that may be even more important. So like I have this very deep interest in anti-inflammatory strategies and looking for prevention of autoimmune disease conditions that now affect 20% of Americans with 100 different autoimmune disease conditions, of which type 1 diabetes is just the tip of the iceberg. But we're finding like common path between autoimmunity and strong response to a viral infection, that if you wish are both, they could be counted as both an hyperimmune, hyperinflammatory reaction to a viral trigger. Like in diabetes could be an enterovirus, in COVID-19 can be the SARS-CoV-2. And now I cannot speak yet for another week about the trial with mesenchymal stem cells, but hopefully we'll have an opportunity, since Dr. Antonio Martos is one of the key investigators and movers of this trial, to discuss it in one of the next meetings, because you can start fastening your seat belts, and I think you'll be very happy with what you may hear. I cannot say anything at this point because it's still double blind, but I can tell you that we analyzed the baseline before randomization, and these are only very severe ARDS, acute lung injury, intubated or high oxygen patients. So we're seeing only the mild to moderate or moderate to severe Berlin criteria ARDS patients. But I can tell you that already there are data supporting what I've been saying, because the vitamin D levels are low, extremely low in many patients, and the inflammatory markers that are diet-induced, specifically the omega-6 over omega-3 index, is through the roof. It's like more than 45.5 as average, which means there has been a very good paper on JCI showing on human PBMC, both in type 1 diabetic subject or in non-diabetic subject, that if you have a high level of omega-3, you improve regulatory T cells levels, and you have a better chance of responding to a triggering event of an autoimmune disease, for example. And you decrease your Th17, the autoreactive aggressive effector cells. But most importantly, if you have high arachidonic acid, or a prototype in this index for omega-6, we measure the arachidonic acid over EPA ratio that is now available even at Quest or everywhere. If you have arachidonic acid elevated, you lose completely your Tregs. Your Th17 goes through the sky. So you may be, just with a simple American diet, Western diet, or lack of protective factors in your diet, be exposed to triggering events that create an hyperimmune, hyperinflammatory reaction, where in a person that is healthier, diet and supplement wise, you may not see this. And I think this will be very important. Nobody's really interested, because most of these molecular supplements are not billion-dollar molecules, so you will not see the hundreds of million investments to develop them or study even them. But we started a collaborative network with Italian groups at different universities. And hopefully we will be able to speak, Lillian, I'm looking at you specifically, and others that may be willing to join the resistance of studying things that may be non-profit based. So on one side, we are doing the mesenchymal stem cell trial as a non-profit repository, thanks through support from foundation to provide a cost or no charge cell doses. But on the other side, this project we're doing in collaboration with this Italian group is called the Batman project, or Fit for Pandemic, which means how can we shift most of the population towards the asymptomatic or pouch-symptomatic mild cases, so that you try to avoid the need of intubation. If you have a severe cases, we still have mesenchymal stem cell or whatever will become proven as effective. But if you're not before, how can you prepare the population to be more resistant, like bats are resistant to any coronavirus or viral infection, and could serve as a common strategy both to address the pandemic of autoimmune diseases or the pandemic of viral diseases. So I think that will be a very interesting project for any of you interested to learn more or join. I'll be happy to.
I think that's fantastic. And I agree with you. The key has to be, we need therapeutic strategies for the critically ill, ARDS. What do we do when you are super sick? And then what do we do with prevention? The people in the middle, more or less, we get them to fly. So I'll be happy to talk to you.
Thanks.
Okay, thank you, Camilo. I would like also now to ask Dr. Sandra Rizzoli, as editor of the Panamerican Journal of Trauma, Critical Care, and Critical Surgery, to quickly describe what kind of papers has been submitted to the journal, and what kind of research have you been seeing around the globe. And then after Dr. Rizzoli, I'm going to go straight to Dr. Abu. Dr. Abu, you can summarize this meeting and give some messages to the globe about lessons learned so far and the future, please. Right. Dr. Rizzoli, then Dr. Abu.
Okay, so I just would say Dr. Martos, Dr. Abutanos, and all the presenters and discussants was excellent. I really enjoyed it. Thank you very much. So this is an unashamed invitation to consider submitting your work to the Pan-American Journal of Trauma, Critical Care, and Emergency Surgery. If you're doing something in trauma, COVID, and critical care, COVID, and emergency surgery, especially for a young investigator starting a career, consider the Pan-American Journal as a possibility to put your work. We look forward to it. We have an expedite review. We're reviewing papers now in about 14 days, and it's fully peer reviewed. There's no phase that's being omitted. So thank you very much for the opportunity, and consider the journal. Thank you.
Thank you. Dr. Abu, you have the podium, please.
Well, first of all, I want to thank Dr. Martos and Dr. Abutanos, because this is the 20th anniversary. It's a big accomplishment. When we had the first one, we thought it was just going to be 3 people on a call, and just this resilience, and every week you just get better. So first of all, thank you for keeping this meeting. It's terrific, and I think a lot more people hear to the recordings after, and it's really Correct. Fabulous. So first of all, thank you. I think we heard 3 different and phenomenal speakers. Dr. Rapaka, Dr. Doblecki, and Dr. Schulman did a phenomenal work summarizing what's the current state and what's in the pipeline, so we can understand vaccines, the differences, the similarities, and the other clinical trials. We look forward to hearing the results from Dr. Ricordi soon. We hope we can stop the counterfeit of drugs in Bolivia, so Dr. Foyani can get real medications. And I look forward to hearing the results from Dr. De Lima in Brazil, and understanding if really favelas have what we say in Venezuela. They're hard to kill, you know. We would see someone with 10 gunshot wounds from a favela and they make it completely fine, and then you see somebody from a different neighborhood get 1 gunshot and you don't know why that person doesn't make it. So yes, I hear there's something in underlying immunity that we need to study. I think we will know more in 4 weeks than what we know now. And thank you all. Stay safe, wash your hands, wear your face shield, wear your mask, and social distance.
Thank you, Lilian. Thank you, everybody. Next meeting will be in Spanish. Everybody stay safe, and see you back in 2 weeks. Thank you.